So welcome everybody. I'm going to introduce Brandon today. Thanks for you guys attending in person and for everybody online. I'll give you a little background about Brandon. So Dr. Brandon Southall led the NOAA Oceans Acoustics program back in NMFS S&T a decade ago. I can't believe it's already been a decade ago. And he's subsequently conducted extensive research on the effects of noise on marine mammals. He's a senior scientist at Southall Environmental Associates and a research associate with the University of California, Santa Cruz. He's led major research programs on behavioral responses in marine mammals, which we're going to hear a little bit about today, as well as recently leading a major synthesis and review of how noise affects hearing for marine mammal species. And I just want to say, I was fortunate enough to start my career when Brandon was here and had a little bit of time overlapped with him to work with him. And on behalf of all of us, we value you as a colleague and definitely as a friend. So I'm really excited and happy to have you here today, and I'll let you get to it.
That's great. Thank you, Amy. It has been almost 10 years, I think, to the day. Like literally, it was the end of April, 10 years ago, since I worked here, and it is hard to believe that it's been that long, but I see a lot of people that are friends and that I worked with. I know there's people online that I've known since I was here. And, you know, I want to start just really briefly with something that I'm sure is going to sound corny, but being out of the agency and continuing to work with people in the agency and agencies, especially at the kind of political time that we have now and working on these issues, like, thank you. I'm serious. I really mean that. Like, it is, I know it's hard sometimes to work within, you know, a big bureaucracy and to work in the government at times. And there's people outside the agency, myself, you know, included, that really appreciate what you do every day. And I sincerely mean that. So thank you for having me, and thank you for doing the things that you do. So I sort of have three, I keep gaining affiliations. We just started this new, I work closely with the Duke Marine Lab. I'm going to talk about a project that we're doing on the Atlantic coast that's closely coupled with folks at the Duke Marine Lab. And we just started this nonprofit in California called California Ocean Alliance, where we're teaching high school kids how we do marine mammal science and doing some local research and conservation. I won't say much more about it, but it's really cool. I'm pretty excited about it. If you go to caoceanalliance.org, there's some more information about these interactive programs we're doing with high school kids. But I'm going to get right into it because I have a lot of stuff. But I'm going to basically try and do three things. I'm going to go quickly through the behavioral response study sort of background, because there's a lot of people here that are painfully familiar with why we're studying noise and its effect on marine mammals, but I'll say a few things about that. And then most of what I'm going to talk about is this project we have done in Southern California that started in 2010 and really is kind of still continuing. We've had over 30 papers that are published, and you're going to get like the very high level synthesis of like a whole lot of things. Each paper could be its own talk. So you're going to get kind of a flavor of all of the things that we've done with not a whole lot of detail, but I'll try to synthesize where we think we got. And then I'll hint at or give an overview of two specific projects. This one in the Atlantic that's kind of a direct extension of the work in Southern California, and then another project that I'm really excited about that we're just starting to kind of see the results from, that looks at how do you study these things with animals you can't tag. So all these other ones involve tag-related things where we look at individual animals. How do you study behavioral responses in groups of animals that are too small to tag? And how do we look at not only their behavior, but their physiology? I'm really happy that Nick Keller is here from the Southwest Fisheries Science Center. Nick's directly and closely involved in this, along with John Durbin from Southwest. And we'll talk a little bit about that. I'll try to leave some time and we can talk about some of the behavioral, and Nick can speak to some of the physiology. Okay, so a lot of people sort of know this, and I won't belabor it, but basically sound is really important to marine mammals for essentially everything that they do. When people started doing things in the ocean, they realized that physics dictate that sound is the way that you find things and find your way around. So we put sound into the ocean to look for things. We put sound in the ocean as a byproduct of moving things around or building things. And there's an overlap between the animals' use of sound and people's introduction of sound into the environment. And a lot of the questions and the issues arise from sort of, and a lot of my career, both when I was here and since, have been centered around answering these questions of when are there problems, when are there kind of like issues that what are the issues and what is their severity, and then what can we do about those things, recognizing that commercial shipping has global economic significance and sonar has national security implications. How can we modify, understand, and then potentially modify those things to mitigate impacts? I also probably don't need to belabor the point about Navy sonar and some of the sort of drivers behind that. There have been, starting with the Bahamas, the picture in the top there is from the Bahamas in 2000. There have been a series of stranding events related to military sonar, associated with military sonar. A lot of different people from the academic community to the NRCs to the multiple agencies have said, you know, we need some direct studies. There's a lot of uncertainty as to what the impacts are. So the stranding in 2000, the kind of scientific research happened, was going on in the Mediterranean because there was some awareness of the issue there. And then starting in about 2004-05, when I was here at the agency, NOAA and the Navy began discussing, like, what are we going to do to try and understand this, get some direct answers. 2007-08 were projects in the Bahamas that I was a co-investigator on when I was still in the agency. There was this project in 2009 that happened in the Med that didn't really work out for some reasons, and then this project in Southern California is kind of the big kind of U.S. Response to this. There's some parallel work going on in Norway and in with the Norwegians and the Dutch and St. Andrews. There was kind of a parallel project that has some interactions, but that sort of grew out of that uncertainty, overlaid with the fact that NIMS and the Navy were getting sued and up to the Supreme Court at the same time, and the whole regulatory framework that came in around 2007-08, and now the Navy's kind of in that process, and these things have gone from, you know, a need to now directly informing some of these things. So we're seeing already that the results, some of the works I'm publishing are now, they're directly incorporated into the exposure response functions for military sonar for the U.S. and other countries. Okay, so just kind of a background on the approaches. There's, you know, there are valid concerns about sonar, but what I think we've realized in really looking at this is some of these other species. So we've looked at beaked whales, obviously, because the strandings disproportionately involve beaked whales, but there are many other species that are exposed to active sonar all the time that are not, they're not endangered, that are not involved in strandings, but we've started to see, you know, some sublethal kinds of effects that are potentially significant. There was a pretty big review that we did. I actually don't have all of the references on the slides, but I have like 4 or 5 slides that have all of the references for this project, which I'm leaving on here. If anybody on here or online wants this presentation, I'll give it to you, and it has all the references. When I get to them, I'm just going to skip over them. But I'm putting references on that aren't in that list of 35 papers that come out of this talk. But so a lot of the focus has sort of gone from, obviously we want to understand the mechanisms that may be involved in these stranding events, but we want to understand things that don't involve mortality as well. And a lot of the things in this, there's some references in here that are from this project that are getting at, we're realizing context is important. And by that I mean things like what the animals are doing at the time, what the spatial orientation of sounds and receivers are, what the prey environment is for feeding animals. So a lot of the things I'm going to show you are sort of unraveling parts of that context-dependent thing about distance, space, environmental context, behavioral state. And by using these controlled exposure experiments, or CEEs, it helps us tease some of these things apart from one another and identify some species-specific kinds of things in ways that observational or opportunistic studies are limited in their ability to do. both of these approaches have their pros and cons. We are increasing sort of the realism of these experiments, and I'll talk about the fact we've actually gone to working with real ships from experimental sources. They both have kind of pros and cons, but without the experimental control, the opportunistic observations have some limitations. And yeah, there is a need from moving from the original projects in the Bahamas and some of the other experiments that have used simulations to actually using the real thing, what the animals are exposed to from full-scale operations, which we did get to in this experiment. There's just a lot of people involved. This is a big project. I won't go through anybody's name, but you'll probably recognize some kind of key people in the field. I think the only thing to mention here is you've got government agencies, you have academic, you have nonprofits and private small companies involved in this, with a lot of people with a lot of different expertise in engineering and statistics and biology and field logistics and tags. So there's a lot of people involved in a project like this. This is actually not even probably a fully complete list of everybody that's in the field, but it takes a small village of people to do something complex like this. In terms of the species for the BRS, we sort of split it up into deep divers with a focus on beaked whales as the particularly sensitive species that was involved. We actually worked with both Cuvier's and Baird's beaked whales in California. Mysticetes, and most of our work was on blue and fin whales. Kind of the logic there being they're endangered and they're exposed to these signals. They're probably low frequency animals, and these are mid frequency signals, but not much was documented about their responses. And then other animals that are not necessarily endangered or necessarily sensitive to sound from just observations, but that are particularly common. And a lot of the work we wound up doing in the tag study was on Rissos dolphins, and we sort of figured out that the tag-based approaches don't work on common dolphins and bottlenose dolphins. They take the tags off pretty quickly. They just spin until they come off, and if they don't come off, they just jump out of the water until the tags come off. So thus the kind of lead into the tagless study. That was the original logic. I'll tell you some reasons why I think the tags aren't the right approach for those species anyway, even if you could tag them when we get to it. And they make up a lot of what the Navy and the EIS kind of estimated takes for sonar is in these other common species. Okay, so we wrote kind of a detailed methods-based paper that gets into this, but essentially we're using tags, primarily these short duration suction cup tags, D tags, that are measuring fine scale movement and acoustic behavior of the animals. So the sounds they make, the sounds they receive, how they move through the environment in detail. We did some work with longer duration tags that attach by small barbs that have acoustics and have movement sensors as well. Most of the work that I'm going to show you is from D tags. And then the controlled exposure experiments basically involve a before, during, after design, where there's a period where you're monitoring the animal with tags, also with visual observations, oftentimes with passive acoustics simultaneously deployed in the environment. And then a period of exposure, defined exposure, where we're manipulating the source relative to the animals to get to a specified criterion of how the sound is presented. And then a period afterwards where the monitoring is maintained from the tags and visual observations and acoustics. We worked with, the simulated sonar is a simulation of these. I know this might be a little bit small, but what you're seeing here is about 3.5 kilohertz from several different sources. The large ships, the 53C sonars. This is a 53 sonar, C sonar from a D tag. This is a two-tone sonar from a dipping helicopter, which is slightly different. But these are some of the most common Navy sources that put sonar out as a consequence of training. We had simulations of those from experimental sources that we used as well. And I'll briefly, you'll probably see the term PRN in the slides. We used a pseudo-random noise control that is the same frequency band, the same power, but isn't as tonal. It's just noise within those bands. But we worked up to and included those actual sources as part of controlled exposure experiments with real destroyers and cruisers and with the dipping sonars. Okay, there's kind of a lot to it, and I'm going to go through this pretty quickly and try to get to some of the results. But basically, there was a big step change that happened from some of the previous work that had been done that was more like oceanographic research, where you had like one big ship and a one little tag boat or two tag boats that sort of trailed this big ship with big eye binoculars on it. And we switched to a much more mobile, agile, small tag boats lead the whole thing, and then you bring the sound source boat on another platform that's small and maneuverable. And that turned out to be really important. So there was a step change in the overall field methodology that was pretty significant. We also moved away from the original work in the Bahamas, and part of why the med work didn't pan out so well was just this myopic focus on beaked whales. We added in other species to be able to work on the majority of days when they're not beaked whale days. They're very difficult species to study. They're small groups. They're at the surface for a short time. They're hard to see. So we did manage to work with them, but we added in these other species so we could sort of do productive things on days that weren't workable for beaked whales. We just sort of leaned some of these things down a little bit. These are big and fairly expensive projects. I always hear that. We have made them from the first year to the last year of the BRS. They're about a third as expensive as the first year was, so they were able to become significantly more effective. Moving to the real ships was a big step. This is the first project that's made that kind of coordination with the real ship. Some of this work leads into the tag list, which I'll leave for later on. And then, you know, we're deliberately playing mid-frequency sonar to marine mammals, including beaked whales, based in Southern California, which is where NRDC's headquarters is based. We tried to be very open, very clear, and very engaging about what we were doing and why we were doing it. We did public kind of outreach and discussion about that. We made our work transparent. We made sure, even though we were dealing with Navy systems, that everything we did was in an unclassified format. We tried to do things like this, where we presented things and said why we were doing and what we were finding. And that was pretty important, I think, in order to get people on to understand and appreciate why we were doing it, and to kind of reduce people's concerns over it, or at least discuss their concerns over it. Okay, so I'm going to break down the rest of this part of the talk. I'm going to show you highlights of papers that are published. So all of these things I'm kind of highlighting a whole bunch of things, and I've binned them into basically methods and tools, baseline behavior, and behavioral response, because the project was obviously designed to understand the behavioral response. But some of these things, just as a consequence of having a long-term project that's trying to, you know, study. And in the focal species, I didn't mention we studied 10 different species in this project. So tagging a blue whale and tagging a Rissos dolphin, just the tags themselves have some, you know, some evolution that happened. This is a very actively evolving field. So there's some tools that we developed, and then things we learned about what the animals do just naturally that nobody knew. And then the behavioral response was kind of the focus. So I'll try to go quicker through the tools, but there's just some really cool things here. So I mentioned a couple of the different types of tags. Most of this comes from D tags. The panel on the top here shows, this is the calls of a fin whale, and this is the sound. This is a spectrogram on the top panel in color. You can see the kind of a traditional spectrogram, and then we have the acoustic waveform in the next panel down in the black, and then the blue and the green and the red traces show a signal that comes from the accelerometer. So the accelerometers are intended, and the gyros are intended to tell you how the tag is moving, if the animal's speeding up or down. We figured out if you sample them fast enough, you can feel the tag vibrating when the animal calls. And that's a big deal for things like density estimate from passive acoustics, to know what the calling rate of an individual animal is. And if you just have the acoustics, and you have a fin whale here and you have another fin whale right next to it, you don't know which one's calling. The one that it's, you would think it would be that on the tag on the animal would be louder, but not necessarily. And so we've been able to figure out which animal is calling by virtue of the tag shaking. So that's pretty cool. The bottom panel here shows just a whole bunch of dolphins vocalizing, and the method on the bottom basically takes, the tag has two hydrophones in the middle of it, and it takes the angle of arrival difference between the two hydrophones of a click. And if there's an animal that's coming, that's approaching it, and then goes away from the tagged animal, that angle of arrival changes. But if the tag's sitting still on the animal, the angle of arrival should be the same from the click. So you can determine which animal's clicking in and amidst hundreds of animals clicking. So that's just a pretty cool like development that has implications for things beyond BRS. A number of other things, again, each one of these could be a talk in and of itself, so I know I'm giving you pretty high level things, but those methods to it, the development of that method, its application, we looked at echolocating behavior in Rissos dolphins, which nobody had been able to quantify before, development of some methods automated methods to detect things like how often tagged animals are feeding, and automating some of that. Shane's in the back. Thank you for being here, Shane. Shane was involved with us and led a couple of papers that, if you saw that spectrogram, there was like an intense part and then a long trail that comes after it. That's basically reverberation or echoes happening in the environment. And Shane wrote a couple of papers out of that that looked at that reverberation pattern, the kind of echoes that come after that and what that may mean. These intermittent sounds that are intermittent but that persist in the environment for some period of time. And then what the figure here is showing is basically a tool. So if we're trying to say in real time, we have a tagged animal and we want to present sound to it under certain conditions at a certain level within a certain range, but we want to do that in a pretty accurate way, that's a complicated thing to do. The environment is very dynamic. The animal's moving in 3 dimensions. The vessel or the sound source is moving. The Naval Postgraduate School, and I think Tatiana is probably on the phone. Tatiana Margolina from the Naval Postgrad School developed some really innovative and useful real-time propagation tools for us to be able to say, we have an animal here. If we put the ship here, so the whole thing, it's flipping the whole propagation equation around from the perspective of a tagged animal. Usually you go from the source and go outward. This says if I have a tagged animal here and I put the ship, here's what the sound field would look like if the source was in these other locations. And I don't have time to go into it, but we have been from doing that and then saying that the levels we would predict on animals and then getting tags back with calibrated sensors off it, we're within 3 dB of predicted levels consistently, both there from helicopter sonars, real ships, experimental sources, and in the Atlantic with that method. It's kind of unbelievable, but she wrote a paper and we have some things coming out on that. So that has been essential to do this in order to both meet our experimental obligations and also ensure that we're meeting our permit requirements. Okay, this is just kind of a metadata slide of how many and where. So there were 10 species that we tagged. The majority of them were the focus was on beaked whales. Blue whales was our largest number. This is 92 tags deployed on blue whales, 31 fin whales, 39 Rissos dolphins. The beaked whales are obviously offshore. This is the Southern California offshore range around San Clemente Island. A lot of the blue whales are coastally, but you still have some blue and fin whales offshore, and then the Rissos dolphins sort of spread around, a lot of them around Catalina Island. And then on the right panel, it shows the total number of CEEs that we did, which is over 100 within these 4 species, and there's some smaller number for some of the other species, but I'm going to focus on those 4 with about 20 instances where we have a real sonar at a known location, tagged animals in a controlled exposure scenario from the real ships and the helos. Okay, in terms of the baseline data, and I apologize for going over this pretty quickly, but I want to get to the response stuff. three different kind of examples of this. The panel on the left shows some survey track lines from Jay Barlow's group. Tina Yak was the lead at that time, Jay Barlow at Southwest, that identified some new unknown sort of beaked whale habitats that have been the subject since of a lot of survey effort that has proven to be a pretty good habitat area for beaked whales. And then just from, you know, these are the first high resolution tags on Cuvier's beaked whales in the Pacific. The first ever tag that's ever been deployed on a Baird's beaked whale, like just how do these animals dive? How deep do they go? What do they do out of some of these? The sample sizes are small, but these are the first measurements that we have of those kind of things. Okay, this really could be like almost a course, the amount of work that's gone into like the energetics and kinematics of feeding in blue whales. Jeremy Goldbogen, who's in town this week at the ONR Review, is a professor at Stanford. His lab looks at dynamics of movement and kinematics of feeding in blue whales, and there's been a whole bunch of cool stuff published on it. The top panel is really complicated, but I'll basically just show you. The red line shows the total aerobic dive limit that would be basically predicted of what animals would do if they were trying to stay within their aerobic dive limit. The circles here show what we measured in terms of feeding, in terms of the number of lunges as a function of depth. And this blue line basically represents what you would do if you're trying to maximize your energy gain, which means sometimes exceeding your aerobic dive limit. So it just demonstrated essentially optimal foraging in blue whales, which hadn't been done. And then Jeremy's work is looking at a lot of fine scale movement behaviors. We demonstrated some flexibility in lateralized strategies. So whales have lateralization. They're basically handed, but they can kind of go against that in instances where they're near the surface, and they can probably visually utilize things. So they tend to roll they tend to roll more to the left when they're near the surface, and more to the right when they're at depth, which just has to do with neural architecture, and just shows all of this stuff collectively shows much more dynamic foraging behavior in blue whales. People sort of think of them as just like mowing through the water and munching things, and they do not do that. They're very dynamic and very agile in how they feed, and there's a lot of stories behind this. But so this is just, you know, again, things that come out of this that don't have to do with the behavioral response. Some similar patterns in fin whales. There's a paper about to come out on this. I won't go over it in detail, but basically fin whales optimize their feeding, and they forage where the prey is, and when it's easier, like near the surface, they forage more than they would relative to the density of more than they should relative to the density of prey. So they're basically balancing the available prey at the cost of foraging, is the bottom line method of this. There's some work done on Rissos dolphins. Patty Arons, who's also here this week. The part of this that I love the most, there's a paper called Rissos Dolphins Planned Foraging Dives, and I won't go into it in a lot of detail, but the basic message is they search on their way back up from one dive, and what they do on their search pattern on one dive up dictates what they do on the next dive. They're basically feeding an environment that has 3 or 4 different prey layers, and they search the local environment and then plan their next dive based on what they found on the previous dive, which is probably really common but not well documented in the animal kingdom, or at least in the marine environment. So a lot of cool stuff on that. Okay, so kind of really getting into the meat of the response studies. So there was an important paper published by Stacy DeRuyter. This shows the dive pattern of a beaked whale, tagged beaked whale. The light blue is echolocation or clicking behavior. The bottom is, and I don't have time to get into the details of the statistics, but it's basically integrated 5 different measurements of movement behavior in an integrated distance-based statistic. And when the animal goes down, the exposure happens on the second deep dive, and almost as soon as the sound goes on, the animal stops echolocating, has a shallower than normal ascent, and a long series of surface dives. It does start feeding again or starts echolocating again on the next deep dive, but there's a long series here, and all of these behaviors basically change quickly at the onset of that, and it takes a while for that to ramp out. So we can see that there's a clear change that affects both its horizontal position and affects diving behavior. The panel on the bottom is with the Baird's beaked whale. It's a little bit harder to see, but all the way on the bottom, essentially this is movement intensity or acceleration. The animal picks up speed and about triples its speed, and this response intensity metric goes up as well. So it actually goes away a little bit quicker in the Baird's beaked whale than the Cuvier's, but these were kind of our first two sort of documentations of these responses in beaked whales. The blue whales is probably the biggest data set. We have 42 blue whales that were exposed in different scenarios with where we could tease apart, and this is a pretty important finding, where we have animals that are not feeding, animals that are feeding near the surface, and animals that are feeding at depth. And the top figure here, they all show before, during, and after, and this is a general additive mixed model approach that basically shows there's no difference in diving and feeding behavior, in diving behavior before, during, after for non-feeding and surface feeding, but there's a large change in the deep feeding animals. They disproportionately change their behavior more. The bottom panel is a little bit complicated, but it's basically a behavioral state switching model that uses hidden Markov models. And if we take essentially these three different states of travel, surface feeding, deep feeding, what we find during exposure is animals that are not feeding don't start feeding, and there are basically four different types of whales, or whales that have certain types of behavior as they're being exposed. The surface feeding animals don't start feeding, and the deep feeding animals the ones that shows up sort of the strongest, and the difference between them, these red squares down here, the deep feeding animals are more likely to switch into not feeding. So it's essentially showing the state dependent change. This is a little bit more obvious when we look at the individuals within individuals, and we say, for all 42 of those, was there a change in that individual's behavior? And then we pool those across all individuals. This paper was just published about 2 months ago in the Journal of Experimental Biology, and we used two different methods. One of them was this Malinobis distance, which is the integrated statistical method. The other one was called expert scoring, which essentially is a way of utilizing people that know the behavior and biology of these animals and saying, is there a change according to this specified criteria, this specified severity of behavioral response? The methods turn out to agree pretty well with one another. I personally think the expert scoring methods are a little bit, it's not just what do you think, it's experts in multiple groups pooling results according to specified criteria. It's a little bit smarter. The Malinobis distance is just sort of, did this behavior change from a standard distribution? But the methods pretty much agree, and you sort of see this. So here's where we get into the PRN, but which actually is interesting that we're seeing responses to the pseudo-random noise and not just the sonar. So it just tells you it's not something that's just specific to the characteristics of the sonar. It's sound in that frequency band that's influencing the behavior of some of the animals anyway. But the big contrast here is the deep feeding animals relative to the shallow feeding animals, where we've got 3/4 of the deep feeding animals responding and none of the shallow feeding animals. The other thing that's important in that, if we look just at the deep feeding animals, and this is hard to get your head around, but there are three different panels on the bottom here that say 1 kilometer, 2 kilometers, 5 kilometers. So this is saying for if the source is 1, 2, or 5 kilometers away from the animals, and these have the same axes on the bottom that go from 110 to 170 decibels, these are probability functions that say what's the likelihood of an animal responding for these identical received levels. And if you look, and there's two different types of severity responses represented, but if both of them show the same pattern. If you just look at the top, this is the probability of responding. So 50% probability of responding at 140 decibels at 1 kilometer, whereas at 2 kilometers, that's 130 decibels, or for 50% would be actually, you don't even get to it with the midpoint, and then a much less probability at 5 kilometers. What that tells you is the animals are ranging. They can identify not just how loud the sound is, but they can tell how far away it is from them, and the farther it is away from them, for the same receive level, the response probability is decreasing. They're using methods of ranging. We have some ideas about how they're doing that. Probably has to do with Shane's reverberation patterns. It may have to do with harmonics. It could have to do with onset. So there's that, I think, is one of the main findings that we got to out of this, is that these animals are using things other than the loudness of the sound to dictate their response, which has some important implications. I think I'm gonna just preemptively say there's a paper coming on calling behavior where we're not actually seeing. There's been some previous work published on blue whales that indicates that cessation in calling is a function of sonar exposure, which we're not seeing in this study. There's a lot of details there. I want to make sure I get to the tagless study, so I'm going to kind of skip over this. But again, I'll give people, anybody that wants this presentation, I can give you the details of it. I think I'll say the same thing about fin whales. So there's several papers still to come. What we're doing with the fin whales is the blue whales, and we kind of argued with ourselves about, do you wait till the end and publish it, or do you have papers that come out? With the blue whales, we published them along the way. So the one method came out, then the hidden Markov model method came out, and then the expert sporing Malanovas paper came out. With the fin whales, we're doing all of those in one paper, which is kind of a beast of a paper, but it's still to come. The sample size is half that of blue whales, so some of the patterns don't look like they're as clear, but with that caveat. So that's a paper. The calling paper is still to come. The fin whale paper is still to come. And then this is probably, in some ways, the most important paper that may come out of this, which is, these are all the instances where we had a real ship or a real sonar. So these are all of the exposures that where we used either directly with the incidental, where with the CE, we have a few incidental exposures in there. But the thing to take away from this is, and many of these animals include, the blue whales include deep diving animals. There's only 2 instances. One of them was kind of a questionable type of response at a pretty low level, but kind of a low confidence, minor change in diving behavior. One of them was a very clear response, and this was dipping sonar at 146 decibels. But other than that, in none of the other instances where we had real sonars at 10 kilometers, 20, 30, 40 kilometers, we didn't see any changes, including the Beaked Whale. So there's a Beaked Whale CE on here. So we're up to 125. That's well above the level where we had these really clear responses when the sonar was 5 kilometers away. Now we have the ship at the same level, 30 kilometers away, and we're not seeing any change in behavior at all. So that's probably a pretty important paper to come, but that's kind of where it's leading, and I think it's this range dependent thing that's getting us there. So closing out the BRS part of it, in terms of the methods, there was just a lot of development, technology development, experimental methodology development, the baseline behavior, new species that had never been tagged before, diving, calling behavior that hadn't been documented, and then on the behavioral response side, getting at these context-dependent differences, behavioral state and range dependencies is pretty important. These are things that are already being incorporated into the exposure response function estimates. And I think really this insight into context dependencies of state, prey-dependent variables that I didn't talk about a lot, and range are substantial findings that make things difficult for people that are trying to develop exposure functions, but are a consequence of just how things are. I think if you think about it, what level of sound bothers you, and then you put yourself in a library when there isn't a talk going on versus a busy restaurant, those are 2 different answers, and that's probably what's going on with the animals as well. Okay. The range receive level issue is really important to tease out. I think it has major implications for how you set rules over and estimate impacts. There's some work that's going on in that regard. We're trying to do a little bit more. And then the tag development, moving to long, these are short exposures. So I didn't mention that the exposures are 30 minutes or an hour. These things happen for, you know, hours to days sometimes. So getting tags that go over longer time frames is something that's needed and is happening. Okay, here's all the papers, which I'm not going to belabor, but I just left them in there because I had them in there. And anybody that wants this, you want all the references, they're all in here. Okay, I'm going to go quick through the Atlantic project because I'm really excited about this tagless project. I'm excited about the Atlantic project as well. Don't get me wrong, Andy, if you're listening, Andy Reid, he's probably not on the line. But the other project is really cool, and Nick is here, and I want to talk about some of the stress hormone stuff. So, but this project is kind of the sort of, and within the Navy, it also moved from the Living Marine Resources kind of 6.4 research into the fleet monitoring program that's supporting it. So it's significant that it was sort of a transition into more of the monitoring realm. There's still research components to it, clearly, but it's kind of the most direct evolution of this. So we're working with the fleet out of Norfolk. In this case, the Duke lab is kind of clearly leading, you know, a lot of the field work. They've been doing work off of Cape Hatteras for a long time, and there's some reasons why it's a really good place to work. So one of the criticisms about some of the work that's been done, like in SoCal or in AUTEC, is that in the Bahamas, is that they're on ranges. They're in places where these animals hear sonar like daily, you know, or often, commonly. Cape Hatteras is in, and then there's been work that's been done in very, very remote places like Jan Mayen, north of Norway, where there's no sonar at all, and animals respond very strongly. But if the Navy never really goes there, what relevance does that have to them? It has relevance to sort of identifying the bounds of that. Part of the reason we picked Cape Hatteras is it's in the middle of those two things. It's a place where the Navy goes sometimes, but it's not an active range. It's also a really high density place. This is, you know, one of the richest environments I've ever been in. You know, you don't need to be a biologist to appreciate the richness and the hotspot nature of this place. That just the water changes immediately, and you start seeing birds and turtles and fish and fishermen there quickly and in localized places. But there's been a lot of success in tagging Cuvier's beaked whales. There's a lot of pilot whales around there that kind of give us that option when we can't work with beaked whales. We don't know what the responses of animals off the East Coast are. And I think from a lot of these things we've learned, we're now asking in the Atlantic, we're asking pretty specific questions like, how does species receive level, and then some of these contextual factors of influence behavior? And we're actually asking the questions with those things in mind, as opposed to sort of figuring them out like we did in SoCal. There's a lot of transition, so we're working with essentially the same design. However, getting at that longer extended duration, we're simultaneously using the short duration D tags as well as satellite tags to cover hours to day, weeks to month kind of duration simultaneously. So there's two different tag types to try to get at space and time. Yeah, D tags, and then primarily splash tags with dive data on them. I won't go into this, but this HiCom model relates to anybody that's been off of Cape Hatteras knows it's an extremely dynamic environment that can change from day to day, where the currents are, where the eddies are. You have to take that into account in terms of the propagation analysis. We're actually using Navy models on a daily basis to change that the propagation problem based on local conditions as reported through buoys and XBTs and things like that, which is phenomenal that we've been able to do this in this really dynamic environment. The other thing, and I'll show you one slide on it, but it won't do justice to it, but Rob Schick at Duke does a lot of work on animal movement modeling, and there's a lot of error in these satellite tags, so you have to actually account for that error in estimating, and they don't have acoustic recorders on them. So we're accounting for spatial error in the tags and uncertainty in the animal movement in 3 dimensions, and making predictions of receive levels using animal movement modeling and estimates of spatial error. And Rob and Tatiana from NPS are really involved in some of that. And we're asking really specific questions. Instead of like, is there a change? We're saying, is there avoidance? Is there change in foraging behavior? And are there changes in social behavior? So we're tuning our questions and tuning our methods to kind of get at some of these things. Again, I really won't get at this in sufficient detail, but basically, if you take the data that you get from the satellite tags and you run a whole bunch of simulations that say, here's 100 different interpretations of this filtered track, and then from those, then this just illustrates those points before, during, after for one of those 100 tracks. But from those, you create an uncertainty polygon of where you think the animal is, and then you do the receive level modeling in defined depth bins. You can now have a statistical representation in 3 dimensions of what the predicted receive level is from a satellite tag that has spatial error in it. Never been done, and kind of insufficiently represented in other studies with satellite tags that just take 1 point and 1 depth. This is kind of comprehensively characterizing error and uncertainty in receive levels, which is a cool evolution. So far, we've put out 55 satellite tags, 38 of which are on Cuvier's beaked whales in 2 years. So those of you that have been following any of the beaked whale deployments in other areas, that's more than anybody else has deployed kind of anywhere over any duration. So not to brag, it's just it's a very dense area of beaked whales that's there. So there's been a huge amount of baseline data. We've done 10 playbacks so far involving 105 discrete kind of exposed events, known events. 3 of those have involved surface ships, so actual 53C sonars. We've deliberately done these deliberately and a function of the fact the animals move themselves, but we have ranges from 1 to over 100 kilometers with both tag types, not the long distances with the D tags. But and I'm a little cautious here with what kind of the results are because we're still in the middle of it, but we haven't seen animals like ceasing to use the habitat or moving for days, as has been observed in some of these really kind of pristine habitats. They continue to use the habitat, but we're seeing some short-term kinds of changes in diving and some avoidance behavior in some, but not all of the animals. We're working next week. We have someone going on the water to start putting satellite tags out for some effort in May. We're continuing for a couple more years, and then there's a lot of work going on to kind of continue to improve the analytical methods. Len Thomas's group at St. Andrews is kind of advancing some of the analyses. Okay, I took more time than I wanted to, but so bear with me for just a few more minutes because I want to talk about this project to look at responses where we can't tag animals, right? So now we're talking about oceanic dolphinids, common dolphins, bottlenose dolphins that occur in sometimes groups of thousands of animals. How do you study these animals, right? They're just difficult, and people haven't gotten much on them. Like I said, they haven't been involved in strandings, but they're common, and we would like to get that right. So this is relevant to the Navy. So we came up with basically a pilot study to try and quantify both fine scale and larger scale movement behavior and different aspects of behavior, acoustic behavior in these animals that are fast, that change their behavior a lot and occasionally occur in large groups. So it sort of looks like this. There are things that people have been doing. They're not brand new methods, but putting them together in this way is pretty new. So, and this is John Durbin's team that's flying octocopters, and I know that Dave Detler and SMT have been very supportive of John's group, and John wanted to make sure we acknowledged SMT's role in helping to support the photogrammetry work that they're doing. Shore-based theodolite surveys and high-power binoculars, both from shore and also from boats. Obviously not the theodolites from boats, but visual sampling. And then we use drifting acoustic recorders. It's a little hard to see, but there's a drifting recorder here with a bunch of dolphins going around it. So we're fast deploying single hydrophones where we can get them in really rapidly around animals. And we learned a lot about how to do that and how not to do that, because you can attract common dolphins really easily if you drive in front of them with any kind of a boat. So we figured out ways to be able to do that. So we're trying to get them around them, but also not influence their behavior. We're using the same so far we've used the simulated sonar source, and then we're using conventional biopsy sampling methods to get at some of the physiology. Focal species are primarily common dolphins, but we have done some work with bottlenose dolphins. The Rissos dolphin work is kind of intended there to sort of overlap with the tag work, where we can look at some comparisons there. And most of the work, well, all the work has been around Catalina Island. There's 2 different stations, primarily this one off of Wrigley and this one on the back side of Catalina. But we can, sometimes the animals are not close enough to shore. I've been impressed. They've been able to track animals that are 5, 6, 7 miles offshore, up to about that range. Sometimes they're not in that proximity, and we have to kind of pull the visual observers onto vessels, and we've done that. This is an example of the theodolite-based work. So here's the observation station. The animal with the red cross, the animal about 8 o'clock, we start tracking the whole group. They come down here. The red part of it is during the exposure. I'll show you the sound source is over to the northeast of here. I'm not sure there's a map of this one, but the animals in this instance actually kind of go towards the sound source during it. But we're also measuring detailed aspects of behavior, number of clusters, cluster size, spacing, distances between clusters, shape of the whole group, whether it's all in a field or a line abreast, behavioral state. The animals actually slow down slightly during the exposure period, display events. So we're getting both the big picture movement as well as fine scale aspects of behavior. Okay, I'm going to go to this one. We're going to step out for just 1 second. I'm going to show you 3 quick videos that start with a bird's eye view. So this is, you're in the UAV, the UAS, we're about 120, 150 feet above the animals, and you're looking straight down. You can go ahead and play it, and I'll just kind of talk over it. So there's actually audio associated with this, but you're not going to hear a lot until you hear the first ping. So you were looking straight down. These pictures are taken once a second. You can see this whole field of dolphins there. Hear the reverberation. You can hear the echoes. So this is in real time. So the UAS is straight over the animals. The recorder is about, I think this one's about 600 meters off the center of the group. So in this instance, the animals, from just kind of the orientation perspective, we don't see like a big orientation change here, and we don't hear a large kind of acoustic response. That's the rep rate's 25 seconds, which is modeled after what the Navy really does. Okay, so that's just that's one view. I didn't want to show you the extreme one first. So they don't always respond, but this one you'll see a clearer response. And listen, let's see if we can turn this up maybe just a tiny bit. Can you hear the whistles going on? Sorry. Hear the whistles pick up. So in that case, and the sound source is off to the top left from them. So that's a case of, and that's the first ping. So it's a 10-minute sequence with 1 ping every 25 seconds or 24 pings. We actually turned the sound source off after a couple pings of this one, because the response was so clear and strong and sudden. So that's an example of a pretty strong response. And then this one will sort of lead back into the methodology a little bit. So this is a bigger field, and if you look closely, like you'll see the dolphins, but there's a yellow line that goes along with each one. This sort of demonstrates how we get to the statistics I'm going to show you next. So for every frame, every dolphin in the frame is marked head tail location in real lat long space with centimeter resolution. So every individual in the frame, granted you can see sometimes the animals are going out of the frame, but every animal in the frame is marked in real space. So John's UAS has very precise, the error resolution of the device is inside the device, and it has a laser altimeter. So we're really measuring animals in pretty fine scale in space. Okay, so from that, we get things like these cloud plots where, when we come back, you'll see all of those are stitched together, these 1 second representations of each animal that's in the frame is represented here with little arrows. So the orientation of these are 1 second frames. All of the position and orientation of all the animals is represented in real space. And this just shows that track that I showed you. So the sound source was off to the east-northeast. The animals came up here after the third ping. They slowed down, went over towards it, and then kind of turned away from it during the exposure. So from that looking at it in terms of directionality, thank you, Aaron. We can, if we take 25-second windows that sort of represent each ping, and we look at directionality, these are histogram, 360 histograms that show what is the relative orientation of every animal in the group. So this is the before. Here's the first ping during, and then in the third ping you see them turn to point towards the source, and source subsequently. So we can get fine scale resolution of directionality within the group, and we can put that in the context of the whole group. So now we have statistics of all the individuals that are in the frame represented in terms of directionality, and the one on the right here is change in spacing, so the relative change in the pairwise spacing of every animal in the frame, which is amazing. I cannot believe that we've been able to do this type of fine scale information. I cannot say enough about John Durbin and Trevor Joyce and Cassidy and Keiko at Southwest that have done this. I think it's beyond what I envisioned we would do with this pilot project. Just a couple of quick examples. Sorry, I'm running a little long here, but I'm almost done. This shows another instance where there was a pretty strong directional turn. It didn't happen until the third ping for this individual, but there's a pretty clear change here. And this is on the top right is a spectrogram of like just the instance right before and right after the first ping. So you can see in the instance you heard, the animals were already calling a little bit, and then they just had this step change. Here they weren't calling at all, and then you hear this crazy cacophony that happens. I won't talk about the way we score the acoustics. It's complicated. We've tried some automated methods, but when you get into that, like, try telling me how many animals are there. We have a greater than 50 category. but in terms of using multiple scores, looking with sort of structured automated methods, when we see this type of change across all those phases, we can statistically show a clear change and increase in acoustic behavior. Okay, plug for Nick's work, and if people can stick around, maybe we'll give Nick a second to talk about this a little bit afterwards, Nick, or we could take a question or something. But just the Nick is, and we're in the field collecting biopsy samples at defined. So this is minutes post exposure from 20 to 200, and just three sampling periods. The blues being controls, and the orange and reds being cortisol levels following exposure. And a longer term function of Nick's work that I know one more slide on that looks at opportunistic sampling over a very large area, kind of gives these baseline levels for blubber cortisol. These are our controls. These are very similar, obviously, in the opportunistic versus the control. And then we see this increase, significant increase in the blubber cortisol post exposure. So we have not only behavioral changes, but physiological changes as a function of that. And Nick can talk a little bit more about this, but they're sampling animals from Point Conception, Monterey Bay, south of Monterey Bay, all the way down to Baja, and finding, getting these baseline blubber cortisol levels. And then just wrapping up this part, I think we proved that we could do this, which was definitely a question. This was really like a difficult thing that I wasn't positive. I think we were all sort of wondering whether we could really do. There's a lot of baseline data that we, including some things I didn't even show you, like length histograms for thousands of common short-nosed and short-beaked and long-beaked common dolphins that is relevant for other kinds of reasons. I think, like I said at the beginning, we started this because we couldn't keep tags on these animals. Having watched these animals and used these kind of methods, I don't think tags are the right way to do it. Unless someone's gonna develop a really, really tiny tag you can simultaneously put on 400 animals. I don't think so. It's like birds. It's like a flock of birds or a school of fish. the group as the unit of analysis. And so these kind of integrated methods to study them, I think, are the right way to go about it. We do have another phase of this project, but this wasn't a kind of a dedicated behavioral response study. It was a pilot study to see if we could do some BRS. We've seen some instances where there are changes, clear changes, some where there aren't, and we have another phase of this to add some sample size, focus on some specific things. We're starting to see some potential state dependencies, but it's a little too early to say, I would say, with the number of samples that we have. And the next phase of this is planned to incorporate some real sonars as well, just the helos, not the real ships. But that's the next 3 years. We actually just got ONR support to extend this study. And then I'm kind of ending with I'll put all these up and just sort of talk generally about this. And really a lot of the focus has moved from the stranding events into sublethal effects. These context dependencies that have been revealed are really important and need more work. The integration of behavioral and physiological effects is a really positive direction. I'm glad that we've come together with Nick and his team. Moving to longer duration and realistic scenarios is important and happening. And taking these things and feeding them directly into some of the population level assessments. ONR is just supporting some PCOD work to take the behavioral response study from blue whales and work it directly into PCOD models, and that's the right direction for some of these things to be going to. And I think with that, and 10 minutes later than I hope to be, I'll stop, and if you guys are willing, we'll take some questions. Thank you.
Okay, so we're going to take about 10 minutes for questions, and we'll pass any online questions we don't get to on to Brandon as well. So, any questions in the room? So, you did have a few come in.
Yeah.
With your blue whale results that you were trying, what's the depth for the shallow feeding versus the deep feeding whales?
Yeah, we used 75 meters, which is a little bit arbitrary, but there was, there's some deep-like feeding that happens around that depth, but it's kind of a break point based on
75 and shallow.
Yeah, and shallower is shallower. Yeah. There's kind of a hole in blue whale feeding that goes between 50 to 75 and about 150. But it depends. It depends on the bottom depth. But we used a kind of a statistical kind of break point of that. But many of the animals, typically when the animals are shallow feeding, they're surface lunge feeding. So they're like at the surface. And I think, some of the motivation for that is when they have prey that's right at the surface, their ability to they're kind of pinned. They can't go up, and so they've got them in a scenario. They may not. They're actually not as dense or rich as the really deep patches that are particularly valuable to them. And this is why Jeremy and Elliot and Ari's work that looks at the kind of value of these patches shows that deep prey patches are really valuable, but they're much more ephemeral. When they get these surface layers, the whales they may not be as dense or as valuable, but the whales can really get at them. And so I think their lack of response is probably a function of the fact that they're in a scenario where they're able to get something easily and pretty good, and they're less likely to be dissuaded from that by some external disturbance. Whereas if they're feeding on these, ephemeral and distributed patches, and there's a disturbance, they're more likely to move away from it. That's my interpretation of our work anyway. Yeah, Jolie.
Yeah, 2 questions. First one, sorry if I missed this. Did you have a snow level estimate for any places where you started seeing responses with this?
Yeah, no, you didn't miss it. Sorry. I've been a little. I did have it on. No, it's yeah. So the one with the quick turn was in the 140s. They range between 125 and up to 150. So it's in that we don't have any exposures that are over the that are over 150. So they're all in the 120 to 150 range. I've just been a little bit reticent with it because to show it, because we only have, I mean, there are 13 within common dolphins. There's 9.
And how far away was it? So that's obviously.
Yeah, it's around. Sorry, the first one with the theodolite had a plot around 2 kilometers.
So.
Which is, well, let me just say one quick thing about that. Like some of the experimental sonar work before, and as I showed, we had this contrast between the experimental sonar and the ships. The distances between the helo dipping sonars are I think it's much more representative. The experimental sonar is much more representative of the kind of scenarios like that the dipping sonars occur in, where they're stationary. You're missing the rotor part of it, which if you're within a mile or so, there is a component of the actual helicopter that gets into the water, which a ship doesn't have. But at a couple of kilometers, it's indistinguishable because we try to measure it. So it's stationary. It kind of comes out of nowhere, and the durations are more like what the helicopters do. So I think it's a pretty fair representation of those types of instances, and it's about the durations that they typically operate.
So the other thing, if I can remind, that's one question.
Sure.
So I guess I heard you say you had like 9 and 13, so 20-ish applications of the tablets method, and what I heard you say is that the group is the right scale at which to look at these responses. And I guess I'm wondering, so you've kind of seen things that you would generally characterize as the group did a thing versus the group didn't. And the reason I ask is because, in the business of interpreting sort of old studies where there were groups, it's like, oh, well, I see half of them did X and half of them did Y. And I'm wondering if you see any sort of info suggesting that you might parse out they all generally responded, none of them did, sometimes there's a partial response, or if you're really kind of seeing generally, you can characterize it consistently as the group had an adverse response versus.
So,
could you repeat the question?
Yeah, repeating the question. So Jolie asked whether there was whether there was a consistent all the group responded or all the group didn't, or whether there was some mix in that. And the short answer is we do have instances where we see sub components of the group respond in ways that other parts don't. So one in particular that I showed yesterday, that the group the UAS was over continued, that was actually closer to the source, continued on a pretty straight track, and the theodolite group that was the group the theodolite was tracking had kind of an avoidance response. And it was farther away, but actually the modeled levels are higher at the group that was farther away than the group that was closer, which is interesting. I don't think we have enough resolution on kind of any of this yet to get too carried away, but yes is the answer to your question, I guess, about whether they were mixed. Well, what is a group becomes a question with some of this, right? Because like in those plots that I showed that had the behavioral tracks number of clusters, within the course of 2 hours, a group of 400 animals can be one group of 400 or 4 groups of 250, 50, 10, and 10. They're dynamic. They're subgroups that are coming together and they're spreading apart and they're coming back together. The situation I'm describing is one large group of 400 animals that had 50 here and 350 down here. The 350 moved in a way that the 50 didn't. But 10 minutes later, they're all one group of 400. So did the group respond or did a subgroup respond? I think that's why it's important to have both the broad scale and the fine scale to be able to get at. I mean, a lot of what we see in the changes that we're seeing in the group dynamics has to do with cohesion. And when there are orientation responses, typically the animals get closer together.
Okay, and we're just thank you, Brandon. We're just going to have to end the broadcast there.
Okay.
But I will again forward online questions on and continue the conversation.
Okay. Thanks everyone for joining online. Did you have another question?
Yeah, I was just sort of